realize that he was like 75. He looked like a much younger looking person. In any event, again, welcome back. Hopefully we will not have any kind of technical problems most of the second half of the year. Audrey, you ready to get started? The word of the day, I don't know what the word of the day is.
Pennsylvania.
Pennsylvania. Okay. It's all yours. You get that done?
It's on Sunday.
Okay.
So Happy New Year, everybody. To ease us into the new year, to fire up our neurons, we're going to talk about a very light topic of evolution of the immune system. Actually, as I was preparing for this talk, I talked to Dr. Allman. I said, wow, this talk is actually quite dense. And he said, actually, that he was trying to think about who would give this talk. He had a person in mind, but couldn't remember their name. And so, alas, you got little me to help you navigate, take you on the tour of the immune system. So in the first part, we're actually going to be looking more at sort of the evolution of the adaptive immune system. But before I begin, I have no disclosures or conflicts of interest to disclose. And so in animals, in almost virtually every animal system that has been studied, except for the C. elegans, there have been these migratory phagocytic cells. And so you can sort of think of them as like little macrophages or monocytes that sort of ingest and digest the different organisms. And the C. elegans, they don't have a cellular immunity. Instead, they use lectins, antimicrobial peptides, and lysozymes. But the question is, how do they recognize these foreign invaders? And that's not yet known. And then, as we know, there's sort of basic pattern recognition receptors which recognize conserved pathogen-associated molecular patterns, such as what we know in mammalians, the toll-like receptor and the TNF receptor pathways, and these can be mimicked in other earlier organisms, such as in the Drosophila. So there's this theory of this big bang in immunology, and basically there's a change from the innate immune system to the adaptive immune system. And so the adaptive immunity has been first found as defined as what we know in humans to be found in cartilaginous fish. And so it was really thought that, wow, we're this very complex immune system, whereas these earlier predecessors don't have that. Because when we looked at the jawless, which is what agnathans means, lack thymus, spleen, and there were no trace of immunoglobulins, TCRs, or MHCs. But I'm going to show a little bit of how that actually was debunked. So the thought was in this Big Bang that RAGs, which are the proteins, are involved in the recombination of the BCR and TCR, that it was probably induced by transposons. And so going back to your basic biology, transposons are jumping genes. So you may remember in your corn, the variegated pretty corn that you get at Thanksgiving, that was discovered by Barbara McClintock, and she actually won a Nobel for that. So transposons are indicated here in the red, and basically this is foreign antibody that's that can, and they call it jumping gene, but it really isn't a gene. It's a segment of DNA that basically cleaves itself out and then can interpolate into other parts of the DNA. And so the thought was perhaps that this was what's happening and how we got the RAGs. So as you know, there's the RAG1 and RAG2 protein, and here they're showing it as being all on one transposon, but the actual thought is that perhaps RAG1, which is really the recombinase one, is the one that was the part of the transposon, and RAG2 developed much later in vertebrates. But basically, this transposon that contained the RAG protein from some invading organism incorporates into the host DNA, and here they're showing in the green that this may be some genes that are encoding for antibodies. And then basically these RAG proteins cleave that genes with them, and then they go and insert into, sort of clip and pasting into another part of the DNA. And over time what happens is these RAG proteins stop losing its ability to jump, so to speak, and then they just become enmeshed in the DNA. And then instead of removing just any old part of DNA, it helps to bring that dysfunctional or other parts of the antibody gene together to make a functional gene. And then over time, with in generation, you can get gene duplications and sort of divergence of this population. And so this was first hypothesized in 1995 by Craig Thompson. And when they looked, actually, in another group looked at homologs in earlier organisms, this group in 2006 found that there are RAG transposons or homologs in purple sea urchins. But at this point, that's not known to be involved in the immune system, but they're not sure exactly what those proteins do. So just going back and reminding you how antigen receptor gene rearrangement occurs. So as you may remember, that basically there are these RSS or recombination signal sequences, and this helps the RAG proteins recognize to bind to that area to bring the joining either V, D, J domains together by creating a hairpin formation and then cleavage, and then these open-ended DNA segments have to be glued together, so to speak, and so it's used by using endonucleases and other polymerases. This allows for the rejoining of these genes together, and so you'll get these sort of X-sized circles, which just contain the signal joints. And so, trying to think about it more clinically, why these are important, RAG1 and RAG2, if you had the absence of that, then of course you wouldn't be able to get B cells or T cells, but you would be able to get the formation of NK cells, and so that would be a phenotype for skin patients. This slide goes into a little bit more detail about how antibodies are made, and so as you remember, the antibody has two heavy chains and two light chains. The heavy chains are made of the variable diversity and joining and constant region, whereas the light chains don't have that the diversity region. And so there are many different chains that are available in the humans, and so this allows for mixing and matching and widened diversity. So besides having the multiple gene line segments, you can also have the different various combinations which allow for diversity. And then where they join together lends for diversity. And so this figure here reminds you a little bit about enzymes that are involved in that junction, a joining of the DNA. So there's the TDT, which allows for the N nucleotide additions. And then there's sort of the palindromic forms that are made by the P addition. And then in B cells specifically, they have the ability to somatically hypermutate. So again, going back to your basic immunology, you'll remember that in the germinal centers that after the B cells have seen their antigen, they have a very enhanced ability to mutate. So it's about 100,000 times to 1 million times higher than somatic mutation in other parts of the DNA, and that allows for more diversity, and that's mainly in the hypervariable regions, so the endpoints of the antibody. And then finally, together with the heavy chain and light chain, that combination allows for enhanced diversity. So
you just said,
yep.
This may be a naive question. Does all this variability occur randomly, and then these random proteins either have a function or don't have a function, or is it genetically engineered?
As far as we know,
you see an antigen, and then you're hypervariable, and you make a product that has a purpose from the get-go.
Right. So I think it's a little bit of both, but it's so basically sort of like the Burnett clonal theory. So basically, you're making a lot of different kinds of receptors, and the antigen thereby amplifies. So it selects out the ones that are going to be important. So you're right, they're not just going to waste their energy and make a bunch of those cells if the antigen's not around. So as I mentioned earlier, it was really thought that the adaptive immunity was only found in JOTs or higher life forms. But this paper in 2004, the group of Steve Panzer and Max Cooper were the first to demonstrate that you can have somatic generated antigen receptors in animals. So basically those are jawless, so the hagfish and the lamprey. And their receptors look very, very different from in humans, and we use these leucine-rich repeats. And so there are 3 genes that were found named A, B, and C, and it suggests that there may have been a common ancestor that allowed for the adaptive immunity to occur for the generations, and that may have occurred 480 million years ago. These genes for the A and B and C seem to be lineage specific, except for VLRC, which seems sometimes to occur in A cells that have the VLRA, and it's thought that perhaps it doesn't undergo a functional assembly of the receptor and thereby defaults to making VLRC and vice versa. So the VLR gene organization is as follows. So there are the LRR, so these leucine-rich repeats that are on the 5' and 3'. And intervening, there's actually the germ-ine VLR itself does not code for anything. So then by recombination, and I'll show you later on, these LRR cassettes are brought together, and this allows for a functional protein to be made. And it's thought that the VLR genes may be made from sort of mimic the pattern of a platelet glycoprotein receptors. And very similar to in humans, there's a diverse number receptors that can be made. So basically, right here in the lamprey, the VLRA, there are 513 cassettes, and in the bees, there are 820 cassettes. So you can imagine that if you're combining all these different cassettes, you're also where the actual combination occurs, the fusion that occurs. It's not just tight. Sometimes you can have a couple of LLRs being brought together, will allow for more diversity. What the difference is that unlike in humans, there doesn't appear to be any affinity maturation or somatic hypermutation, but you can see that you can have 10 to 14 to the 17, 10 to the 17 different kinds of receptors. So that's pretty impressive. So you can't really say that the earlier life forms are any less complicated or as diverse as us. So this slide shows how the VLR genes receptors are being made. So on the left side, this is what we know in the humans with jaw vertebrates. Besides the RAC proteins, there's an enzyme called the activation-induced cytosine deaminase, or AID, which is very, very important in somatic hypermutation, gene conversion, and also class switching. And so again, trying to make it more clinically relevant, as you may remember, if you have a mutation in this particular enzyme, you can get an autosomal recessive hyper IgN syndrome. And so the B cells are, the immature B cells seem to expand, so you get lymphadenopathy, splenomegaly. These patients seem to be more susceptible to bacterial infection. So similarly, in jawless vertebrates, there seems to be an enzyme that allows for the bringing together of these LLR regions. And the enzyme is called a cytosine deaminase, and so it's very similar as the AID in that it allows for the change of the DNA nucleotide to be switched to uracil, and uracil, as you know, is not one that's part of the normal DNA alphabet. So this allows for a break in the DNA, and so then endonucleases and DNA polymerases help try to fill that break. And by that, how that happens is that when that break occurs, there's a homologous pairing with the LLR. Sorry to touch the pointer here. It seems to advance it. Okay. So LLR modules, and so basically it's bouncing. It's bouncing from from here over to here, and then eventually it bounces back and brings those DNA together. So
Drew, how does she use the pointer on that?
It's the touchpad usually.
Oh, yeah, that's what I'm doing. For some reason,
Yeah,
I touch a touchpad. And
this is a new computer for this room,
so I apologize if I'm making everyone dizzy.
My home computer.
Okay, so the next following slides show how these VLR cells, how they sort of mimic or can parallel what we know in humans. So the VLRB, which is aptly B, is actually very similar to B cells. So the receptors that are being made can actually be found in the plasma and also appear on the cell surface itself. And it's generated in the hematopoietic tissues, and it expresses antigen binding. And interestingly, when they looked at what kind of genes were being made in these VLRB cells, they seem to express very similar ones that we would find in B cells. So PAX5, PCL6, CXCR. So it seems to be sort of a parallel there. Similarly, the VLRA and the VLRC seem to be almost like the T cells in the agnathostomes. And so where they looked at the lining of the pharynx of the gill folds, which parallels sort of where the thymus would be developed in us jawed vertebrates, these are where these cells are created. And unlike in the LRB, it's not plasma expressed, but only expressed on the cell surface, and it seems to respond to classical T cell mitogens, such as PHA. The question is, what does it see? In T cells, obviously, it's an antigen in the context of an MHC. Here, we're not so sure yet as to how antigen is being recognized. Is it recognized by itself? Is it processed? Is it being processed and expressed in a particular different kind of molecule? And then again, they looked at sort of the different genes expression within these cells, and they seem to parallel what is found in T cells. So the VLRC in particular seems to be more of a gamma delta kind of T cell. So as you may remember, that it has sort of innate and adaptive properties. In humans and mice, some of them use canonical receptors so that they're found more like in the skin or in the gut, but they can also not have that necessarily. Some of them also can be recognized lipid antigens. So there may be some of that canonical receptor expression in the VLRC cells as well. Okay, so now we're going to switch and look mainly at B cell receptors. And so the following slides will show how what we know of typical isotypes of antibodies have been shown through different early predecessors. So IgM seems to be the most ancient antibody class, and it seems to, the actual shape of it, how it looks, is pretty much copied pretty regularly through the more advanced organisms. It was first seen in cartilaginous fish, so these are the sharks, the rays, the skates, whereas in bony fish, it seems to be more of a tetramer, and it has ability to change, so it actually, based on reduction or oxidation state, it can change its affinity. And in sharks, it's thought that perhaps it can change from a monomeric to a multimeric state. And so the question is, are these two separate lineages, or is it actually just one cell that's able to switch based on antigen recognition? And that's still not yet known. IgD also seems to be very ancient, but it seems to be more flexible, more plasticity over time. Some species actually don't even express it at all. The function of it is not well known. In frogs and mammals, it seems to be primarily transmembrane, whereas in bony fish, it seems to be more secreted as well as can be bound. And because of studies looking at fish, they looked a little bit more carefully in humans, and it seems that humans also have secreted IgD, and these can bind to the surface of basophils and have very strong pro-inflammatory properties, and so they can release various cytokines such as TNF, IL-4, and IL-13. And some of these studies were actually done in patients that had periodic fever. So as you remember, hyper-IgD is known as one of the genotypes for phenotypes for periodic fever. And so in looking at these patients who have more pro-inflammatory phenotype, this is what they saw was the basophils were quite activated. The IgD seems to be also secreted very much so in our human respiratory tract, and so that they can show that these bind bacteria that are known to cause respiratory infections. And then IgG and IgE seems to be derived from an IgY-like ancestor, and the IgY are still found in birds, reptiles, and lungfish. Interestingly, they seems to be in high concentrations in the chicken egg yolk. Unlike IgG, though, it does not activate complement, and it doesn't seem to bind Fc receptors. There are shorter forms of the IgY that seem to neutralize pathogens without activating the inflammatory response. And then IgA, as we know, is important in mucosal immunity. It's first found in reptiles, and it can be either in monomer or dimer form. In bony fish that don't have IgA, they actually use a different immunoglobulin called IgT, which also is very similar in function in providing mucosal immunity. Sorry, just skip that part. IgT twice. And then in camelids, so in camels, they have IgG, which is very different. So you can see here that they don't have that second chain, so it's only a single chain variable region, so there's no light chain. And it seems to also have a form that's similar to this immunoglobulin mu antigen receptor, which we'll talk a little bit more about later when we talk about TCRs. So when you look actually at the genes of that create the TCR, in humans, as we talked about already, there's the V, D, J, and the constant regions, and they call this a translocon arrangement, whereas in cartilaginous fish, they don't actually have it broken up with all the V's in one area, all the D's in one area. Instead, they call it a cluster arrangement. So you have the VDJ all in one, and then another pattern of VDJ. And so you can still get junctional diversity in somatic hyperpigmentation, but there's no switching apparatus between the clusters. And then amphibians were the first to actually have class switching. And then in TCRs, we have sort of a similar arrangement, but in sharks, interestingly, in their delta genes, in a quarter of them, they actually have this 3-domain receptor chain with 2 variable domains in a constant region, and this is the NAR TCR. And so you can see here that there's this additional vertical region just sort of plopped on top of the alpha at the gamma delta chains. And so these receptors seem to recognize soluble antigen, and the gamma TCR seems to still be able to hypermutate. In the interest of time, I'll just skip a little bit faster. And then looking at the antibodies themselves, as looking at the gene families, you can see more sort of the flexible evolutionary changes that can occur. So, for instance, in IgG4, as we know, we sort of think of this as more sort of like a pacifying antibody. It can activate complement. It has low affinity for C1q and Fc receptors. It has more of an anti-inflammatory activity. What was interesting in my reading, which is actually an older paper, but I wasn't familiar with it until doing this, was that it can exchange Fab arms. So we always think of it, the antibodies, once they're being made, as being fixed. So in this study, they used nude mice, and they injected them with human recombinant-made antibodies. So you have IgG4, which recognizes birch, or IgG4, which recognizes cat antigen, and they also had it in the IgG1 isotype as well. Right. So at the bottom one, with the arrow, the IgG1 for both birch and cat, you can see that the binding, what they're measuring on the y-axis is the concentration percent binding to for both for birch, whereas if you have So if you have a combination of IgG4, birch, and to cat, now you get a 50% binding. So it seems that the antibodies are able to sort of change their specificity. It's not just all to birch and it's not all to cat. It's sort of a mixture of both. And in fact, if you look in humans, you can find they found humans that have antibodies, one single antibody that has a bispecific recognition. So the bottom in B, you can see sort of, if you think about cat being the red ones and the yellow ones being ones that are specific to the birch, the chains actually break open, so to speak, and actually come together so that you get a bispecific, so two different Fab fragments that are being recognized. And when they did looking at which sites would be critical for this Fab RNA exchange, it seemed to be the CH3.
I remember a plenary presentation a couple of years ago at the meeting specifically about this and why it was biologically valuable. That they forgot the punchline. Why is it biologically
Yeah, so
valuable?
hopefully this slide will say a little bit about that. So you can imagine that if you have this, so basically it's kind of biologically relevant. So in the human itself, basically evolving to try to counter inflammatory states, or you can imagine doing it and injecting into a person to potentially help change them and make them less inflammatory. So basically, in this first slide, this is just solely in vitro. What they're looking on the y-axis is the expression of surface anticholinergic receptors. So this is a model for looking at myasthenia gravis, and this was in rhesus monkeys. And so if you have IgG1, so it's an antibody against the acetylcholine receptor, obviously now it doesn't allow for the expression of that surface expression to be observed. Similarly, if you have IgG4 that recognizes the acetylcholine receptor, you'll get diminished surface expression. Now we do it in vivo and ask what happens. So if you have IgG1, which is the antibody that causes degradation of the acetylcholine receptor, now these animals will clinically exhibit symptoms. So all four of the four animals have that. But if you inject the IgG4 antibody against the acetylcholine receptors, those animals did not receive the clinical symptoms. And now the question is, if you combine both of them, what would happen? And those animals survived. So it shows that you can get formation of productive IgG4 antibodies in vivo. So looking at the actual gene for IgG4, the question, one can see that there might be some pressure to have a potential benefit of having more G4 around. So there are tandem duplications of the G4, and the thought is that perhaps in more industrialized Western societies where you don't have as much infection, would you potentially have more of these sort of gene duplication to try to help suppress allergies, whereas in countries that potentially have more parasites, more risk of infection, that you wouldn't have much of a predilection for this G4, IgG4 duplication. And so when they looked at Italians, Japanese, and the Chinese, there were 44% and 22% of the tandem duplication, respectively, whereas in the Gambian population, only 5% had the tandem duplication, but they did exhibit the deletion of the G4. So it's thought that perhaps the genes can evolve to help with the environment based on environmental pressures. Similarly, in IgA, there's a duplication of the gene. So IgA1, which is very important in protecting against protein antigens, unfortunately, the way it's structured, the hinge structure, is very prone to being broken down by bacterial proteases. So you would think that perhaps it would be in the organism's advantage if that hinge protein couldn't be broken down as much. And so, indeed, there is another form of the IgA2, which has a shorter hinge and is more resistant to proteolysis, and these seem to be important in polysaccharide and lipopolysaccharide antigen. So if you do have more cleavage of the IgA1, then there seems to be increased production of IgA2, showing another evidence of evolutionary changes. So now sort of shifting gears and trying to make it a little bit more clinically relevant to us as allergists. Well, why did allergies develop and why did they evolve? And this cartoon kind of jokingly says that, well, perhaps, these little organisms, we're always so serious when we say we do something wacky. Let's overreact to something harmless like peanut butter or pollen. Well, so what really makes an antigen an allergen, and what is the purpose of allergies? So one theory is that allergies are a misdirected response. And so basically, the Th2 responses we know is very important in the first line of defense against macroparasites, so against worm infections, biting arthropods, for instance. And so in these articles by Dave Artis and Finkelman, they said, well, perhaps allergies are, quote, a misguided Th2 cell response, and it's a price we have to pay for effective immunity against certain parasites. And so if that's along those lines, if that's true, if IgE-mediated immune responses evolved to provide extra protection against parasites, in this paper that just came out this year in PLOS Computational Bio, they predict that the environmental allergens should share key properties with parasite antigens. So they collated allergens that are known to cause allergies and or bind immunoglobulin B, and they looked at parasite proteins, specifically the nematodes, arthropods, and flatworms, and they collated them into 10 different superfamilies that we'll talk about a little bit later. And they used computational analysis to look at the actual protein sequences, but more importantly, they looked at the 3D structure, because it's not just how the amino acids are arranged, but how they fold and conform that are important in eliciting the immune response. And then the investigators manually looked at all this printout and said, okay, well, let's look at the topology. Let's look at see if there are certain epitopes or epitopic-like regions that are equivalent between these respective homologous proteins. So half of the parasite proteins from the 31 species fell within the 10 most abundant allergenic protein domain families. And so you'll see in the third column the number of parasite proteins that are detected for the various families, and then the fourth column is the number of allergens. And some of them, they could correlate, whereas, for instance, in the bottom one, the prolamin, which is a plant storage protein, there doesn't seem to be any sort of homologous one found in parasite allergens. But of note, Anastochus, as we know, is a worm that, if it's not well cooked in seafood, for instance, can cause anaphylactic reactions. So this is one of the only organisms that is known to absorb both a helminth and a food allergen.
So before you move on, explain, like, for example, the vet V1 line. What are the numbers 58 and 145?
Yeah, so that means of the parasites they looked at, 58 of those parasites seem to have similar homologous proteins to 145 of known allergens.
And all of those being structurally similar to vet V1.
Exactly. But what we're going to highlight in the next slide, so the EF hand family is a very common motif that's seen in allergens. And in this paper, they excitingly found that it's the first example of a plant protein pollen that's found in the worm. So Schistosoma mansoni seems to have a particular protein, SMVV1L, which is very homologous to B3-4. So this is a unique sort of finding. What we, as allergists, are well familiar with is the tropomyosin sort of cross relationship. So Ascaris or Ascaris is very tropomyosin is very cross-reactive to dust mite as well as cockroach allergens. And so in filarial infected patients, many of them too will have a lot of increased IgE binding of dust mites because of that cross-relationship. So the flip side of why allergies have developed is this toxin hypothesis. And it's actually an older hypothesis that kind of got buried and then brought to light by more recent studies. And it was first proposed back in March of 1991 by this woman, Margie Prophet. And she's actually a very kind of fascinating woman. She doesn't have any formal training in biology. She actually studied math. She was here at University of Washington. But she was sort of like an evolutionary biologist, and she won a MacArthur Fellowship Genius Award for her thoughts on why menstruation occurs. And she actually felt that it was important as part of the immune system, that basically slopping at the lining allows for removal of infection, and basically having that increased blood flow allows for the immune cells to be there. So it was a sort of a novel idea. And she also proposed here that basically allergies are evolved as the last line of defense against the extensive array of toxic substances that exist in the environment. And so that the IgE antibody-mediated allergic response typically causes vomiting, diarrhea, cough, tearing, sneezing, or scratching, may help expel from the body the toxic substance that triggers the response. And so this was sort of a novel idea. And then the group at Yale, that's a Tox group, sort of expounded on this and said that allergies are not a misdirected unintended response, and that it's not just worms. Basically, the reason we have the allergies is to help against venoms, against irritants, against other allergens. And so it's not that all allergens have an obvious relationship with parasitic worms, and that really there's no obvious reason to respond with extreme urgency when dealing with slowly replicating macroparasites. And that in some studies, IgE is not important in fully for immunity to the helminths, but it's important in hypersensitivity. And so this group, the slide kind of expounds on that group's theory. So basically, worms, helminths, noxious xenobiotics, venoms, and hemophagus fluids, so things that are like from the mosquitoes and ticks, and then irritants such as the poison ivy can cause allergies. And so there are various responses of the immune system which allow for defending against these particular insults. So for instance, you have barrier enhancement, so IL-4, IL-13 allow for more mucus secretion, so it actually stops and impairs the entry of these different pathogens, and it may even help block their ability to feed and to grow. And then there's a repulsion and expulsion step. So basically all the sneezing, vomiting, the actual scratching, the physical scratching removes that tick from your body, because it's not just the tick, it's that some of these ticks actually harbor diseases. So you're removing potential infections. And then you actually have killing of these organisms. So either through mast cells, through the eosinophils, through different proteins like such as heparin and proteases. And then you get restriction of these different pathogens. So you can get granuloma formation. So basically, through the inflammation of trying to kill some of the pathogens, sometimes it'll also hurt oneself. And so to sort of restrict that, the thought is the granulomas allow for that so that you're not allowing for excessive wound and damage to occur. And then similarly, the allergy response may include some tissue repair. And so you get fibroblasts and macrophages that help allow for extra the collagen matrix to be deposited and to try to repair that barrier. And then finally, there's actual avoidance. If you've had an allergic reaction, you felt terrible, you're throwing up, well, you're not going to want to do that again. So you're going to avoid that eating or touching that particular particular snake or food. So looking more at the avoidance side, this I actually found this area kind of interesting. So allergens can actually directly activate sensory neurons, so the C fibers, and elicit sort of reflex responses such as the itching, vomiting, the diarrhea, and the coughing. They can also interact through mast cells, so IgE specific binding through histamine, and similarly it can also activate chromaffin cells, and which is lined in the mucosa epithelial, releasing serotonin, and the sensory neurons have the ability to recognize this to allow for this for the reflexive response to occur. When they looked at mice, they looked and saw that actually it's not just avoidance; it's a bad experience, but that is really tied to the sensory neurons. So basically, mice, if you were made to be allergic to ovalbumin, and normally they would like to drink something very sweet. But if you keep putting ova in that sweet liquid, now you can teach them to realize, I don't actually want to drink this. So they call it aversion behavior. So they have these mice that are allergic to the ova, present them with two bottles, one without the ova and one that's excessively sweet and has ova. Now those mice will not favor that sweet nectar, and they'll go towards the flavorless one. And that seems to actually correlate with ova-specific IgE. So the more IgE, ova-specific IgE you have, then you have more of that aversion. And if you block the C fibers, then you will terminate that aversion, and you can actually adoptively transfer the ability to have aversion behavior.
If you can activate the C fibers, can the allergy reactions actually create pain instead of just itching and
Yes,
burning?
I would say yes. So transferred by splenic cells and also by the serum. And then they looked in another study where they had these ova-sensitized mice, and then they showed that their behavior can actually change. So mice like to be in the dark, but if you put in nebulized ova-albumin into a cage which is more lit, now the mice kind of detect that ova-albumin, so to speak, and they don't want to be in the light typically, but now because they basically the ova-albumin is in the dark place, now they're going to favor going away from that and going into the light. So their actual behavior can change, and it seems to also correlate with anxiety and actual changes in the expression of various genes, namely CFOS. So CFOS expression was shown to be in different parts of the central nervous system, and this aversion behavior is again dependent on IgE and mast cells. So if you use inhibitors that block IgE or block mast cell degranulation, now you won't have that aversion behavior. So it's kind of interesting because we have obviously a lot of patients who, I call it the sixth sense, young children who always wanted to avoid eating particular food, and you always think, oh gosh, is it just because they're being picky? Well, maybe not. They actually know something. Their body's been sort of has this aversion behavior created, and they know not to eat that particular food. So the next two studies, actually, it's not this one. So what we know is that mast cells can enhance innate resistance. In this study, they looked at snake and in honeybee venom. So c-Kit, which is important in mast cell development, if you have ones that are c-Kit defective, now those mice are going to be rendered not having mast cells. So basically the colored blocks are the wild type. The knockout is the one that's open, and then they reconstitute the immune system with bone marrow derived mast cells and inject them into the mast cell deficient mice. And they looked at how they would respond to venom from a western diamondback rattlesnake snake or honeybee venom. And on the y-axis, they're looking at the temperature change. So basically, their temperature is going to drop. They're going to be going into more shock, which is also a sign of morbidity. And then they actually look at how many mice died in the chart below. And so, not surprisingly, the wild type seemed to be fine. So 13 out of the 15 seemed to be okay from the venom from the rattlesnake, whereas all of them died if they didn't have mast cells. But if you reconstitute it, now those mice will survive, and a sort of similar pattern in honeybee venom. And they looked, and they saw that the mast cells themselves were actually making this carboxypeptidase A, and that was important in degrading venom. And so then 2 landmark studies came out in 2013 out of Immunity, and these were the first 2 papers to show that the Th2 cell response is actually protective. And so in this first study, they're looking at honeybee venom, and they're using mice, and they injected them subcutaneously with various doses of the bee venom. So 100 micrograms of bee venom is kind of equivalent to a sting, to a sting. So they're doing sublethal doses, so 1 to 3 stings per mouse. And then on day 21, they re-challenged the mice with a higher dose, so 4 times of the 200 micrograms of the bee venom. And they looked at their survival. So basically, the open circles are ones that didn't have any sort of prior immunization, and so obviously if they're challenged, they're going to succumb, whereas it seems to be almost dose-related. The more the mice received pre-immunization with bee sting, then they're able to survive better. And so they looked, and they did this in black 6 mice, which are usually thought to be less allergic in nature, but they also did it in Balcine mice, which tend to be a little bit more Th2 in profile, and they still seem to have this protective response. And this resistance is dependent on IgE and mast cells. So if you knock out IgE, if you knock out the receptors for the Fc epsilon, alpha, and gamma chains, then you're going to abrogate this resistance. And similarly, they did studies not just with the honeybee venom, but also with Brussels viper venom, and saw this similar sort of phenotype.
I think the opposite of the human model, that the higher your IgE in humans, the more we worry about systemic reactions to bee stings.
Right. They didn't necessarily look at level of the IgE, but so that's the question: is what's the difference? Why is it that you get a protective response in one and allergic sort of not so good response in other times?
So it's an open question.
It's an open question. And in the second study, they actually looked at the component within bee venom. So phospholipase A2 is the most potent allergic protein in bee venom, although it only accounts for 12% by weight of the bee venom, but it induces more IL-4 positive T cells than whole venom. And it's a conserved component, so not just in bees, but it can also be found in spiders, scorpions, and snakes, and other blood-feeding animals. And what this enzyme does is it breaks down the membrane phospholipids into arachidonic acid and lysophospholipids. And this induces the Th2 response, and that seems to be dependent more on the presence of the lysophospholipids. So sort of similar as the other slides, they looked at the temperature change after giving this VPLA2 to immunized or unimmunized mice. And so if you pre-immunize them, then you'll have less of that temperature drop, so better survival of these mice. And this is a little different from the other studies because instead of doing it subcutaneously, they actually did it IP, so intraperitoneal. So a little bit different, but the doses are actually pretty similar in both. So unlike in the other study, the transfer of serum from productive mice failed to provide protection from high-dose PLA2 exposure, and that was pretty surprising. And they're not sure whether the IgE levels weren't just as high in these mice, and so that's why you can't transfer protection. But they do know that it seems to be dependent on B cells. So if you have B cells that are blocked, then you're not going to get this protection, and it's also partially dependent on the Fc epsilon receptor. So they did the knockout mice in panel B, and you can see that the knockout mouse now loses that protection.
To just comment really quickly, in the the study by Palm there, so he's showing that there's protection to the bee venom itself. So presumably the IgE is somehow against the bee venom. But he also, in that study, they also gave, ova at the same time to see if it worked as an effective adjuvant. And as an adjuvant, it's causing those mice in the periphery to become allergic to ova. So although there's protection to the bee venom itself is actually also acting as an adjuvant, causing the mice, at least in the periphery, to become allergic to ova. So it's a very complex story that's, and then, like you say, he showed that that this protection was presumably due to antibody-mediated protection, but it's really interesting. I don't know if that's part of like what you're getting at with the evolutionary, that there's an adaptive mechanism that is maybe preventing you from having bee venom anaphylaxis, but what happens if you're introducing something else at the same time?
Exactly. That's absolutely true. That sort of that temporal association of having, and that's the thought of potentially why allergies occur. Like so for food, is that temporally you're being exposed to food, but then some other insult, and so then it's skewing your immune system, and now you see the food again, and you have a problem.
Audrey, while we've interrupted you and getting towards the end here,
Yeah.
actually 2 questions. We've gone over so much already, I forgot in the beginning. Did you tell us at what point in evolution IgE evolved?
That's a good question. We don't really know. I mean, it seems to be in the higher life forms, but whether there's something that's not an IgE per se, but similar in function, yes, because obviously you don't need IgE in order to have some of these protections. So it seems like there might be something else.
Actually, 2 other questions. Are there people who study immunodeficiency in non-humans? Are there similar? Is there SCID and so forth?
I know when I was at Penn, one of the groups was doing SCID in dogs, but I don't know that literature very well, to be honest.
And the last question is, maybe there's no answer to this. Is there 2 different models as to why allergy evolved, toxin versus helmet? Is there a punchline?
No, there isn't. And to me, it's almost kind of a subtle point because they're both saying similar things that right. It's not just there to cause us woes. It's there to obviously for maybe a protective reason. And really, it's just is it a worm or is it not a worm? But it's interesting. So basically, I think this is a good way to end it here. Is what decides what makes the immune system decide that it's going to be a protective response or an allergic response? Is it the dose? Is it the species? Because basically, in this Palm study, when they decrease the dose, because if you think about it, right, you're giving the same amount that you would give to a human. So, but in a mouse, the mouse is much smaller than a human. So if you decrease the dose of the venom, well, you get the same sort of protection, and you don't. It goes down, but you actually don't induce sensitization in those mice. So is it something species dependent? Is it how it's being seen? Because whether it's Q versus IP, and so on.
You want to end it there?
Yes, I
Yeah.
will.
So, we have a question I know on the screen back here. Why don't you just read it out loud? You don't need to. I think it's from Chuck Jackson. We showed that IgD on basophils stimulates IL-4, hence Th2 response, as well as IL-1 and TNF, Th1 markers, cytokines. IgD as a primitive antibody seems to be involved in both Th2 and Th1 pathways. You know when in phylogeny Th2 response mechanisms to extracellular parasites differentiated from Th1 to intracellular bacteria and viruses. Long question here to get the point of the question.
Yes, I don't know. That's what made this talk really hard because I knew that people kind of want to know where things started and where things originated, and it's very muddled still, so we don't know.
It seems to me that IgD has always been a mystery to me. If it's been evolutionarily so early, it's got to have an important function, or it would have died out, and yet we never talk about it having any biologic importance in humans.
That's
Which seems, those two concepts seem contradictory. They can be safe over eons, which must have a very valuable function. And you would have a lot of them. You don't have a lot. Any other questions?
Or it's linked to something that's very
Huh?
important. Or it's linked to something that's very important. You can't get rid of it, like getting rid of the other.
Well, an amazing amount of knowledge in your head. Next week, not that this isn't an immensely valuable topic, but next week, much more clinical, it's going to be Steve Tillis doing a review of everything going on in the community and food allergy. It's just exploded in the last